Can you all hear me? Yes. Wow, I'm loud. Wonder how I turn this down. Oh, okay. So I want to start by moving this away from my mouth. There we go. I want to start by acknowledging the Nearshore Habitat staff because we do pretty much all collaborative work, and so everything in this presentation is in part by the rest of the Nearshore Habitat staff. And also the two people that I work most closely with in the Aquatic Reserves Program, which is Betty Bookheim and Jamie Kilgo, who are here. Today I'm going to be talking about kelp, specifically floating kelp, and I'm giving part one of this talk, which is looking from a regional perspective on kelp in the Puget Sound region. And as many of you know, kelp is a brown alga, a type of brown marine algae that lives in temperate areas throughout the world. And it's known as a foundation species, which means that it's like coral reefs, like seagrass systems, like tropical forests. It's a biogenic habitat that's really unique, and it supports a really diverse community. And so it's especially valued. When I say kelp, most people think about, or when anyone says kelp, what most people think about is this species here, bull kelp, which is a type of floating kelp. We have two types of floating kelp in this region, and I'll start separating out different portions of this region. Bull kelp, and you'll also see that we have giant kelp on the outer coast and on the western straits. These are only two species of kelp that we have in Puget Sound. We have more than 20 species here. The other species aren't as well understood. Some are stalked. They project off the bottom on hard stalks. Some are prostrate. They lie flat on the bottom. Floating kelp actually is less common than the other types. You can see their distribution in those maps from some mapping we did in 2000. And what kelps need in general is a hard substrate to attach to, and light and nutrients. And they tend to be found in areas that are closer to oceanic influence with more wave and current energy. The last thing I want to say briefly about kelps before I go into my talk is that kelps compete, each individual kelp competes with other algae and other plants for space to attach to and light. And they have different strategies. For instance, bull kelp is an annual. It's quick growing. It's kind of early successional if you use a classic forest analogy, whereas giant kelp is a perennial. And so they're out there competing for space in a highly dynamic environment. I'm going to start with a really big picture here because a great study came out last year on global kelp forest change over the last 50 years. And what they did was they compiled 1,500 data sets, and they found their first finding was that there's a small global average decline in kelp worldwide. But the more striking finding is that local stressors and regional trends dominate kelp dynamics. And what I mean by this is if you look at this map, some areas are blue, which means there are gains. Some areas are red, which means there are losses. But it's not a coherent pattern. Whereas if I put a coral reef map up or a seagrass map up, it's more consistent across the globe. So local stressors and regional trends dominate. And the other interesting thing is they mentioned in this study that while kelp has historically been really resilient to disturbance, recent studies are suggesting decreasing resilience, which means decreasing ability to recover from disturbance. And so that's something we're thinking about a lot. So for the rest of this talk, what I'm going to do is focus in on local stressors and local trends. Because one of the things they found in this study, let me zero into our region right here. It's very hard to see. And what I mean by our region is two ecoregions. The Oregon, Washington, BC coast, where they had about 26 surveys, and they found stable. You can kind of see that it's pink. And then for the Puget Sound, Georgia Basin ecoregion, they had 2 studies, only 2. And so they didn't classify it at all. So not much is known in our region about kelp. Perhaps it's stable. But luckily, there's a lot of good studies happening now. It's exciting, and so gaps are being filled in with recent research. So what I'm going to do now is go into recent research from our region, and I'll try to specify whether I'm talking about outer coast, Salish Sea, because that really matters. And then I'll also talk about what we know about regional trends, both long-term and short-term. So we've all assumed for a long time that climate drives kelp abundance. And this graph shows that relationship. the connection statistically. The bottom, you can see total bull kelp canopy along the Olympic Peninsula, and on the top, you can see a series of climate indices. And what you see is that, and I've color-coded the climate indices so that the top part is red, which means warm and low upwelling, and the bottom part is cold and wet. And what we found was that this makes intuitive sense, but it's finally, the connection here is supported statistically, is that kelp abundances are lower during El Nino type periods, warm, dry upwelling, and they're higher during La Nina periods, which are cold and wet. So if you look at this, it's almost like a little bit of a mirror image of the kelp and the climate indices, but it's not a perfect mirror image because climate only tells part of the story on what's happening with abundances of different kelps. Temperature and nutrients. These are two of the most discussed variables in terms of kelp conditions. Kelp thrives in lower temperatures, and it requires high amounts of nitrate to grow. It grows very quickly. If you look at this figure from California, you can see that there's a relationship between nitrates and temperature. Basically, as temperature rises, water stratifies, and the shallow waters become depleted of nitrates because they're taken up by plants and algae and other processes. And so in places like California, they've actually defined temperature thresholds where they say thresholds for healthy kelp conditions are less than 14 or 15 or 16 degrees. That's kind of the range that they talk about. This basic relationship has been also made with data from Tatoosh Island so that temperature and nitrates are connected. However, remember, these are upwelling systems. So as soon as we come into Puget Sound, Salish Sea, the oceanic processes are different. And so while temperature and nitrates are important, we can't make the same assumptions that we do in places like California or in upwelling systems. So a few more comments on stressors or drivers. Kelp declines have been measured in response to pollution. This is one of the most famous beds probably in the world, or definitely in the US. It's off Palos Verdes, Los Angeles, and you can see if you follow the black line that in the 40s, this bed went to basically zero from an abundance perspective as Los Angeles was rapidly urbanizing. The population was going through the roof. Sewage was going straight into the ocean. And then as sewage treatment came online, which is the dotted line, you can see that the kelp recovered. There are also documented cases like this in Europe and Australia. But there also, urbanization can have more subtle effects. That was a really extreme effect. Some key impacts of urbanization can be low light, and we heard a lot about the effects of runoff this morning. Another key effect is sedimentation. Kelp attaches to hard substrates, and many of you know that kelp has a microscopic part of its life cycle. We have the big plant that we know, and then on the right of this slide, you can see the microscopic or gametophyte phase. This is the female on the right, and the left is the male, and the little tall thing coming out the back is a baby sporophyte. It will grow to be the plant you know. This phase is particularly sensitive to abrasion, burial, etc. And the other thing that sedimentation can do, which is really interesting recent research, is it can trap particles, and so it can give a competitive advantage to turf algae, which is something we might be seeing. Okay, this is, I think, really important for a place like the kelp beds I study and for Cherry Point. Research has shown that the non-native species of sargassum, which there's lots of at Cherry Point, outcompetes native kelps. So this study that was done by Kevin Britton Simmons in 2004, let me see if I can use the pointer. What he did was he experimentally removed sargassum from sites in the San Juans. And what he found was that if you look at the black triangles at the two sites, you see when he started removing sargassum, the black triangles where the removal were happening increased greatly. That's your density of native kelps and other brown algae. whereas the places he didn't remove it, your open circles, very low. So this is really good evidence that sargassum does compete native algae. And he also did a part of his study that talked about the ecological significance because he showed that sargassum is less palatable to native invertebrates. So there is ecological meaning too. It's not just a species replacement. The left-hand slide shows a site that I've spent a lot of time with in South Puget Sound. That's the south end of Squaxin Island, and you can see the sargassum intermixed in the Nereocystis. I would love to try experimental removal project there, but I don't know if we'll ever get around to it. It's really hard to imagine removing a species like this successfully. Okay, grazers. As you all know, sea urchins are famous grazers of kelp. And sea otters are the classic predator of sea urchins. This is one of the most amazing studies. It's more than 20 years on the outer coast of Vancouver Island. And what Jane Watson did was she looked at 3 places for more than 20 years. The first place, these open circles over here, had otters absent. And you'll see on the y-axis, there was a variety of densities of urchins, but very little algae. The black dots had continuously otters established. There were otters there the whole time. You see very little red urchins, nothing on the y-axis, but a wide range of algal densities. The third graph, as if she hadn't done enough, was they found a site and monitored it for more than 20 years where otters went from rare to established. And what they saw was, at the beginning, they saw the same pattern. At the beginning, you had little algae and a lot of urchins, but a high variety. And as it went through time, it transitioned to a place where you had few urchins, but a wide variety of algae densities. So the message that their conclusion was that you can either have urchin-dominated or algal-dominated phase states. And if you're urchin-dominated, you still have a wide range of urchins. And if you're algal-dominated, you still have a wide range of algae. But it is a fundamental phase state. And in Washington, we don't have high densities of urchins, as you see in a study like this, in part perhaps because on the outer coast we have abundant sea otters. And in some of the inland waters, especially the straits and the San Juans, you have a really active fishery. So I'm not used to seeing high urchin numbers around here, although there have been some slight changes recently, perhaps with sea star wasting disease, which sea stars eat urchins. So it's slight changes, but she actually goes into more detail and looks at all of the different algae. And if you love algae, which I know you do, it is really worthwhile because she goes to the point where she tracks Pterygophora, which is one of the stalk species, and she ages it. And she finds Pterygophora that lives up to 16 years. And she sees it living and in groups, and that it tends to senesce at about the same time. Amazing study, if you love algae. Okay, one more grazer that I want to talk about that we're really concerned about here in Salish Sea, so the inland waters, and that's kelp crabs. If you hang around bull kelp beds, I don't know if you've seen, there are a lot of them. And Katie Dubkowski in the San Juans just finished a great study where she looked at kelp crab feeding preferences and feeding rates. And what these slides show is they love Nereocystis. They love bull kelp. They ate it preferentially compared to both native algae, Alaria and Saccharina. And also down below, they ate it preferentially to sargassum, the introduced species we were just talking about, and to Agarum fimbriatum, which is unpalatable native alga. So she also did caging experiments where she would cage bull kelp so that it couldn't be eaten by a crab. And she found that caged bull kelp was it was gaining mass, whereas the open bull kelp would lose mass when it was available to be eaten by kelp crabs. So that's a really interesting study that gives us a little insight into this grazer that we've been focused on. Okay. We're going to shift topics here and now talk about long-term trends in general. First, long-term losses. This map shows some places where I've heard from people they're concerned about losses in kelp. And what you'll see, there are a lot of yellow stars, is that they're all in the inner reaches of the Salish Sea. And there's a lot of concern about this, and in fact, in 2014, we started a small informal group. Most of these, by the way, are citizen reportings, with a great logo that someone drew up to look at these losses and think about them. I'm going to talk about two areas here. I'm going to talk about the straits, where we don't have any evidence of loss, and then go down to South Sound. I'm going to use long-term, large area floating kelp surveys that we do at DNR. Here are some pictures. Basically, we take low tide pictures. We classify the center picture, which is color IR, and you can see the kelp jumps out as red because it has high reflectance. And then we classify it as the canopy, which is the area that's covered by plant, and then the bed, which is both the canopy and the gaps. We compare these to historical charts. There's amazing historical information. And while kelp canopy isn't the most precise measure in a lot of ways, what's great about it is you can go backwards through time. These are 1911 and 1912 maps that were made to guide harvest. And they're also navigation maps that I've been using as far back as the 1850s. So this next study focused on the rig map that I'm showing you there, the kelp map for harvest. And what we did is we looked along the Strait of Juan de Fuca, which you can see, look at the yellow boxes, and we compared we have 25 years of kelp monitoring there, and we compared the modern abundances, which are shown in the little orange dots above, 25, maybe it's 26 dots, I can't remember, to the historical abundance, which is the one black dot. So that's what Rig found in 1911 or 1912. And you can see we ruled out some areas because we felt the historical data was bad. They're clear. What we found was, and by the way, comparisons like this can't be highly precise. But what we found is, in most cases, you can see the black dot falls within our modern measurements. So it's vaguely within expectations. The one place where things look really different and a conspicuous pattern jumps out is over here in the eastern Strait of Juan de Fuca, where you can see the black dots are consistently above all of our modern measurements. So we don't have evidence of loss, but this is the area that we have the greatest concerns. Yeah, Michael.
Yeah, I'm sorry to interrupt, but out at in the Western Strait, you have the commingled beds of macrocystis and nereocystis. Are you differentiating those two species?
We are. For this study, we did not, because Rig did not. But we actually do differentiate them from the air. And you're absolutely right. From the Western Straits and the whole outer coast, macrocystis is a really big part of the floating kelp canopy. Yeah, Rig didn't differentiate them either. He just lumped them all. Okay, here's a place where we've measured losses. 75% cumulative decline in shoreline with floating kelp present in the South Sound. This is really alarming to me. You can see in these maps that I've subsetted by year increments, year intervals. This is based on about 15 historical charts from the 1850s forward. That the red areas were where kelp occurred. And you can see as you step down, the red's disappearing. So at this point, in the last decade or two, we're limited in South Sound. Here's the Tacoma Narrows, to beds along the Tacoma Narrows, and then 3 beds in the rest of South Sound. And one of those beds wasn't present this year. The Briscoe Point bed wasn't present. So this is one of the areas where we're really concerned. And if you remember that chart I showed you, this is one of those areas in the inner reaches of the Salish Sea where other people are also reporting concerns. Okay, I'm going to shift to recent trends, and this is really cool. I don't know if you all heard, but there was a crash in floating kelp populations in Northern California from the Bay Area north in 2013 and 2014. And you can go to this website, it's at the bottom, but they describe a decimation of the kelp forest, and they have that great logo that talks about it's due to sea star wasting disease, warm temperatures from El Nino. You remember the blob? And what they found was that in 2014, the total area decreased by more than 90%. So a big decrease. And they looked at these sea surface temperature maps show you it got really warm. So more recently, this was in January, they actually started experimentally removing purple urchins to see if they could help kickstart the recovery, because the fishery has collapsed for red urchins. So, okay, so this is what happened in California. What happened in Washington during this same time period, right? Well, let's see. Luckily, I'm very happy about this. We started monitoring kelp, canopy kelp, in the reserves in 2011, which is something we found a reason to do because we could justify it because DNR had set up the reserves. So I'm going to show you trends for all those areas on the left for this same time period. What you see on the coast and the strait, coast is mustard, is we saw about a 50% loss, big loss. It happened in 2013 and 2014. In the strait, we also saw a huge loss, same magnitude, but it all happened in 2014. Different from California, though, we see a strong recovery here, right? And some of you may remember who watch kelp beds that in 2015, the abundance of kelp increased greatly. So unlike California, we've seen recovery here. So here's Smith and Minor Island. We see the same drop at Smith and Minor Island in 2014, and then we also see recovery. And then finally, the part that will be most interesting to you is at Cherry Point and Cypress Island. And note that the y-axis, I've split it. So we've got really big numbers at the top, and then the bottom slide goes from 0 to 20 hectares. And because these are smaller, successively smaller areas. And what you'll see is Cypress Island, which is in the San Juans, saw a loss and recovered, whereas Cherry Point saw decreases, but we haven't seen a recovery in 2015 or 2016 at Cherry Point. I don't have the 2017 data. I think things have improved. You'll hear from Eleanor in a second. But we have been looking at this and saying, scratching our heads, what's the pattern here? And one of the things I'm really interested in is we've downloaded, and now we're doing time series analysis of sea surface temperature data from AVHRR satellite imagery. And you can see I've outlined the reserves. And the sea surface temperature pattern really taught me something that you all probably already know, which is that Cherry Point, if you look at it, Cherry Point's up in the red zone. It is clearly part of the Salish Sea, Georgia Basin. Whereas the other areas that recovered much more quickly are extremely well mixed. They're in the blue lower temperature zones. And again, I talked at the beginning of this talk about temperature as a complex measure. I'm not sure we can use it as a proxy for nutrients. We have to look more in that. But we do know that it is a stressor on kelps. So this is what I'm really excited about working more with. It sounds like perhaps we see the recovery at Cherry Point in 2017. But we haven't worked up all of our data yet, so I can't tell you for sure. So in summary, kelp dynamics are dominated by local stressors and local trends. Where you are really changes the story. And in Washington State, over the long term, it appears stable, closer to the outer coast and the western straits, but we're concerned about the extreme reaches of the Salish Sea, so the farther you get from the ocean. In the short term, we saw that same drop in regional abundance in 2014 that they saw in California, and we've seen a spatially complex pattern of recovery. And finally, I'd like to add a caveat to this, which is our long-term, large area floating kelp data, it's a big picture. It's not really highly precise. So what's the strength about it is we get to compare to historical maps, we get to compare areas and see broad patterns, but it should never replace higher resolution data. And that caveat is important because every different data set has its strengths and weaknesses. And now, Eleanor.
While you're changing over, I could answer
Thank you for asking that question.
Sorry.
It's a great question, and actually what we've seen is our data, I don't have it right here, but I could send it to you, shows the classic pattern that you would expect, which is that Nereo is much more variable, and Macrocystis is a perennial species, so it'll live 2 to 3 years. Nereocystis is an annual. It just lives 1 year. And our Nereocystis numbers are much more variable. Our Macrocystis numbers are much more stable, but both follow the same basic trajectory when you do a cross-correlation analysis.
The other thing about these beds out in the western strait is that they're actually a 3 or 4 species commingled bed, because the Nereocystis is actually attached to Pterogophora, not to the bottom. And so you get a variation of Nereocystis, Pterogophora, Macrocystis, and it's a 3-species dynamic.
So one of the great things I love about the paper I summarized by Watson and Estes is that dynamic, which is Pterogophora is an incredibly long-lived species. This is a species that will live more than 10 years. And so those dynamics in kelp beds and all the algae are really important, where you're getting, there are small-scale disturbances, they open up a little space. The opportunistic species come in, and they're slowly replaced by the more long-lived perennial species until there's another disturbance. Yeah, that's great.
Thank you.
Thanks. Okay.
I'm somewhat terrified this is going to be really loud. Okay, how is that? Excellent. All right, so my name is Eleanor Hines. I am on the Whatcom Marine Resources Committee from now on out. You'll hear me say MRC instead of the Marine Resources Committee, and there are several Marine Resources Committees throughout Puget Sound as well as on the outer coast. But right here I'm talking about my experience with the Whatcom MRC, and then I also work for Resources for Sustainable Communities and do a lot of citizen science stuff. So I encourage you, if you are interested in getting involved, I'll have a slide at the end on some opportunities to get involved in citizen science, but please let me know, and I can probably find a project that would interest you. So I think Helen pretty well covered why we care about kelp, and that we do care about kelp, and that there's a lot that hasn't been studied on it. So the MRCs, the Northwest Straits Commission, who helps to manage the MRCs, came to the MRCs and said, hey, how do you guys feel about helping us collect data on kelp? So most of us said, sure, that sounds interesting. I love an excuse to get out on the water and go kayaking. So it's also another way to ground truth some of the other methods that are out there that I can talk about a little bit later. So when we go out to do these studies, we look at temperature, depth. We do a GPS of the perimeter. We take photos. Each time we take one away from the beach, to the left, to the right, and on the beach, so that we can look over the years at how things might look different over time, or if something weird was going on. Like this summer, there was a lot of forest fire smoke during some of our surveys. And we also do drawings and take observations of like what is going on. Like this year, we had a lot of jellyfish for some reason. And then we also used to look at the density, but it was deemed to be a little bit unsafe, which I'll get to in a little bit. And then something that we're hoping to develop is a health index that may or may not be included in citizen science, but we're sort of discovering whether or not it's appropriate. So our, I guess, 3-year-long journey, I guess 2015 through now, I'll quickly bring you through that of how we developed this. So the first year, we thought, great. The Northwest Straits Commission has this protocol, and we're all ready to go, and we were set and excited to get out there. And then we decided, oh, wait, where are we going to go? Where's the kelp? So this is the 1912 map, or part of a 1912 map of that you saw earlier, where kelp was found. And you'll notice that Bellingham Bay is sort of, there's not much going on in Bellingham Bay for kelps. So We eventually settled on going out to the south end of Lemon Island right there. And on that side, right over there. So that's where we went out. It took a little while to learn the protocol. The rope that we used to take the depth measurements did not work out well. It got all tangled up in it. We found a much better method the next year. So we only had 3 of us out there because we really wanted to make sure that we understood the protocol so that we could teach it to other citizen scientists before having them be a part of this experiment and have them tangled in her depth ropes. So there were also a few glitches that we had to work out. Some of the GPSs kind of had us like, this is supposed to be the perimeter and it's kind of squiggly. Some of the, when you took it, the perimeter showed you kayaking up on land and stuff like that. So there were a few things to work out that we improved. Also, it was a 3-day paddle trip out to Lemmy Island, which, you know, you have to take safety into consideration when you're doing such a long paddle with citizen scientists. And Kim, I think we're going to go a little over. So year 2, we thought we knew better. So we searched for beds with a little bit of easier access. We refined the protocols a little bit. Like I said before, they took out the density protocol, and instead we started drawing pictures and kind of taking notes on how the kelp beds looked. We also, because it took so long to get out to do our surveys, we went out in a motorboat and we took GPS points of some of the other kelp beds around Lemmy Island. And we also GPS some of the points around the south end of Lemmy Island when we were out in kayak so that we could think about at a future point maybe going back out there and doing studies on them. And oddly enough too, some of these beds here, year 1 were non-existent or weren't big enough to survey, and then the second year they were much larger, and then some of the other beds like over here were not there at all. So it seems like there's a lot of different things happening just within a year's time, but we weren't able to measure all of them. So yeah, we noticed some changes. Year 3, I feel like we're getting there and excited for next year. We got to more beds than we ever got to before, so we got to do about 4 different sites. We trained more citizen scientists with easier access to the sites. It's easier to get people out on the water, so we were able to involve more people that didn't necessarily need to have the kayaking experience it takes to kayak across a big crossing and kayak. I think it came out to be about 25 miles round trip by the end. So that was exciting to get more folks out there. And we were also able to do beds more than once with easier access. So it was nice to be able to see the differences just within a month too. And we also started exploring the use of alternatives to GPSs as a citizen science sort of alternative. Not everyone has a GPS unit. So we've been looking at Strava and using Garmin watches and things like that and how accurate they are. We haven't yet been able to compare how it compares to our GPS, but we'll see. And then something that we started to look at was the utility of a health index, what that health index would look like, what we would look at, and if it's something that we can send citizen scientists out to the field in the future to look at some of that stuff. So that's, I guess, coming up in the future. So what's next? I don't know. We'll see next year. I'm excited to go back out to these places and see what we can see has changed from past years. So these are the 4 sites that we went to this past year. We have Gulf Road / Cherry Point, Alden Bank, Eastern Preserve, and South Lemmy Island. So I already told you a little bit about this long paddle. This is basically the journey that we took out there. It takes a while. There were kelp beds out there. It was interesting the first year. We did we got together with the other MRCs and sort of compared notes on how our surveys went. And it was really interesting how much it varied from some people had really dense beds that went on for miles, and you couldn't possibly paddle the whole perimeter of some of the beds because it would take too long. And then our beds were kind of sparse and not nearly as dense, at least that we came across the first year. But I'm kind of finding that that might be changing, especially as we do more sites. So we also found that the kelp beds kind of went straight up to the shoreline here, which can be a safety concern for kayakers, especially if it's super wavy out. And then, this year we went out to Alden Bank, which is, if you guys haven't been there, it's 5 miles from Cherry Point and 5 miles from Peytos and Susha over there. So we didn't want to go kayaking all the way out there. It gets pretty rough. When we did go out there, somebody in our party actually did get a little seasick, and it wasn't like a terribly rough day otherwise. So Bob Cecil was kind enough from the MRC to take us out on his boat, and so we could survey the area out there. Because Dom Mumford, who has studied kelp for a really long time with some other folks in this room, he really wanted to see what was going on out there. And so we got out there, and this is early June. I don't think I was able to take a single photo from the kayak with the horizon line being level. It doesn't look like it's that choppy out, but another thing that we discovered too in doing these surveys is the weather conditions can really change your results. When it's choppy like this, it's really hard to see the kelp. It's better to do at +1 tide or below, because sometimes, like we notice at South Lemmy Island, we sat down and had lunch after we did our survey on the beach, and as soon as the +1 tide hit, all the bulbs went under the water. So we wouldn't have been able to survey them after that, but we found with the other MRCs, it kind of varies site to site. So out here at the +1 tide, it was also the beginning of the season, so we didn't find that big of a bed, and I'll show you actually a comparison to later. And this was actually taken by a motorboat when Bob was out there again, and he was kind of passing through the area and went, "Oh, hey, what is that out there?" And he got closer and went, "Oh, it's the kelp bed that we tried to survey before." So this is also at a +7 tide, and it was, I think, a slack tide. It was close to slack. So you can see the water is a lot calmer and it's a lot easier to see. So it was much, much larger in late September. So we're kind of, I think, hitting kind of the two ends of the season here. So the blue is the one that we got the first time around, and then the yellow is at the end. So you can get different results depending on what month you go in. So the South Lemmy Island site, we try to go back the same first weekend of August every year, so that we're hopefully hitting kind of similar conditions each time and not getting, a smaller bed or a larger bed because we just sampled at a different month or something like that. There's also Easton Preserve, and so that's part of a larger Marine Resources Committee project where there's this quarry out there, and with the Lemmy Island Heritage Trust and the Northwest Straits Foundation, they're trying to restore and enhance the area. And so this is just one of many monitoring projects out there. So plenty of other citizen science opportunities there. So we're looking at the kelp beds here, and it'll be interesting to see how they may or may not change as they redo that shoreline right there. And as I mentioned before, there seems to be a lot of jellyfish out there when we were doing our surveys in July, August this year that we hadn't seen in past years. And so moving on to Cherry Point. So this map is from DNR, and I would agree with what Helen was saying before, where the kelp beds, so we surveyed kind of around this area, and I'd say what I saw this past summer, there was more kelp out there than is shown on this map. So the kelp at Cherry Point this past summer looked pretty healthy. There was a lot going on out there, lots of fish swimming around and crabs and jellyfish and other stuff, and herons on calm days hanging out on top of the kelp waiting to eat fish as they swim by. So we were also lucky this year where Greg Ritter from the Island County MRC was able to fly over the area and take aerial photos of where we surveyed by kayak. And so this is where there's sort of this overlap between some of the other methods of looking at kelp versus actually being out on the water and looking at what's there. Because a lot of times when you take those aerial photos, you kind of think you're seeing certain things, but you're not always quite certain. So this is the photo that he took in August. August 21st, so like kind of late August. We also surveyed at the beginning of August and at the beginning of September. So Suzanne Scholl, who is here and can probably answer more questions on this if you have any questions. She works for the Northwest Straits Commission and did all of the GIS work. So she took the aerial photos and georeferenced them and put them into GIS and was able to auto-delineate what the kelp was. So in purple, that's where all the kelp is supposed to be. And so she was able to look at also our kelp surveys and kind of see like, okay, that kind of makes sense with what we're collecting. So she was also able to measure what the area was. So she got 23 acres of kelp. And then on August 8th, we got about 24.3 acres. And then on September 4th, we got 25.4, according to our perimeters. So the other thing you can see too is our perimeters aren't quite exact. It's hard to see all the bulbs when you're out on the water, and sometimes the weather conditions aren't the greatest, and kayaking close to shore can get a little sketchy. There were a bunch of rocks in here, and I think, I went back a different time for ocean acidification sensors to swap them out, and somebody in our party did get stuck on a rock. And luckily, it was calm conditions and we got him off, but so there's, safety concerns. And kayaks don't always have the best, especially if you have a sea kayak, getting that turning radius around every little nook and cranny isn't always possible, but it's definitely interesting to be able to look at all of them and compare all of them, especially with the image data too. So we're looking forward to experimenting a little bit more with this and maybe looking at some of Helen's data and comparing our results and seeing what we can do moving forward to find more questions and find more answers. So some of the things that you guys can do if you want to find out more information, the Northwest Straits Commission hosts this website. It has a story map, and so you can scroll through here and it gives you like a whole story about how we do the surveys, how you can get involved. It also has some of the data, and so it links to something called Sound IQ, where you get these layers and you can pull up different stuff for the different counties and find our data there. And so with that, I encourage you all to get involved. The North Sound Stewards is a citizen science program or volunteer with the Marine Resources Committee. And you can email either myself or Austin Rose, who is our staff support from the Wacom MRC. So, yeah, with that, I think I'm done. Okay. And I think we have about 7 minutes for questions, and you can ask me or Helen. And if you could please speak into the microphone when asking your question, I would appreciate that. Yes, Michael.
Yeah, very interesting, Eleanor. Thank you. You sort of answered this, but I was wondering both from you and from Helen, what is the coordination between the aerial mapping and kayak mapping in the kelp studies? Because traditionally, the aerial mapping is ground truth. Mm-hmm. So I see the kayaking is kind of ground truthing, but I'm not hearing that.
Yes, sorry, I meant to say that, but I guess I didn't.
Yeah. So you're ground truthing the aerial?
It's a little bit of both. So the MRCs they sort of, it's hard to say what's driving what because they took those aerial photos, I think, because we were studying that area, but there have been cases on the other side too. And I think right now we're really, and maybe Helen can add to this, or she might have something different to say, I don't know. But I think like we're really exploring what, it's kind of like with the plastics, trying to figure out what works best and what different methods are telling us, because each method sort of gives you a different picture of what's going on. And if we only did one method on its own, we probably would be missing a whole bunch of other stuff.
For aerial photos, you're probably aware of this, but just to remind you, I have aerial photographs, 1 each equals 400 of a foot shoreline every 4 years starting 1988. And you clearly see kelp in that, and they have been used for delineating kelp beds in the past. So those are available to you. They are in public domain, and they are available to you.
I think that's part of what's next in year 4, so thank you.
So, is part of the ground truth thing making sure that you're not seeing sargassum rather than kelp?
I would say that's part of it. When I saw the first aerial photographs, my first fear was like, I've been there and I know that there's a lot of eelgrass that kind of overlaps exactly where the kelp beds are. And so it's nice to be able to be out there and kind of tell what the differences are and then compare here. At one of our past kelp workshops, we did comparisons to like using drones and photos from drones versus kayaks, and there's pros and cons to both. The drones take very specific weather conditions. If it's too sunny, you can't see the kelp well enough. If it's not sunny enough, you can't see it. If there's any wind or reflection on the water, it's hard. But you get a nice aerial photo and you don't have to use as much manpower. So there's pros and cons to all of it, I guess.
What was your data? Did it cover the circumference of the bed, or did you deal with individual bulb counts?
So we didn't count the individual bulbs. So part of this too was to make sure that we're all collecting data the same way between all the MRCs. So we looked at the bulbs, and there's a certain bulb density and distance between bulbs that qualifies it as a bed, or the end of if a bulb is more than, I think it's, I forget. I think it's like a kayak length away, basically. Then it's considered a separate bed. And so we would look at the bulbs, and the bulbs were sort of what we used to do the perimeter of the bed, not like the blades or anything else, but the bulbs themselves.
Yeah, 2 questions about sedimentation. One was there a decline in kelp around the Elwha with all that sedimentation, and also since people are suggesting that shoreline armoring deprives areas of sediment leaving coarser stock, do you find a higher correlation of kelp in areas with shoreline armoring?
So, okay, first question, Elwha River. I had something on the Elwha River in this presentation, and I had to pull it because I had too much stuff. But the Elwha River is a great example of sedimentation. And Ian Miller's here, and we just finished a huge study on the effects of dam removal on sedimentation. To summarize, when dam was removed, the millions of tons of sediment that came out led to what we called kelpocalypse, because there was virtually no Nereocystis, no bull kelp in that area. So it disappeared. And what's interesting is it disappeared during this larger regional, it disappeared at the beginning of this larger regional trend that I was talking about related to perhaps temperature and climate. And it started to recover also within that larger regional decline. So we saw really fantastic results there, and we generally believe that it was due, we were asking the question, is it due to turbidity blocking the light, or is it due to abrasion? And it appears that it occurred so quickly and so far away from the mouth of the river, we believe it was turbidity in the water column, so it was primarily light. The second question about shoreline armoring. That's a really interesting question. There's one study that Ron Thom did where he suggested that shoreline armoring may have led to a switch from eelgrass to kelp off Lincoln Park. There's not a lot of, I understand why he suggested that. There's not a lot of strong evidence to either support or refute that. And one of the things that's really bizarre about shoreline armoring is we see really close links. There's a paper that Megan Detier led that I'm like the last author on a couple years ago. And we see a tight link to backshore, between shoreline armoring and backshore and midshore processes, but the link gets less and less strong, and so we don't really have one at this point. We haven't discovered one or measured one. Doesn't mean there's not one, to eelgrass and kelp and the subtidal species. Oh, shoreline armoring is a word that describes any kind of human anthropogenic building in the backshore or the midshore. So a lot of times it'll be a seawall, a lot of times it'll be riprap, or in some cases in highly urbanized areas, it'll be the entire intertidal, like Seattle.
I got a question for you, Helen, if I have time.
I feel like we're cutting into Ian's time.
The question is about your sea surface temperature slide. It showed the northern, or in Washington, the northern part of the Strait of Georgia being really warm. What's going on there? I mean, it was even warmer, I thought, than the South Sound.
One of the weaknesses of the satellite imagery is that the basins, and once you get past Central Sound, the basins aren't wide enough to collect because the pixels are so big in satellite imagery. So there's actually no data in, south of Central Sound on that map. But you're right, and it's just because the satellites can't handle the narrow channels. But you're right, one of the big lessons that I'm having these days, and I don't know if you all were aware of it, is that Cherry Point really is part, temperature-wise and water body-wise, it's much more part of the Georgia Strait than the Strait of Juan de Fuca, San Juan's really highly mixed systems. And that was just something that, I don't work around Cherry Point much, and so this has been big news to me. I'm sure a lot of people are really highly aware of it, but that's one of the revelations I've had.
There's a very good text called Biological, there's very good text from about 15, 20 years ago now, which is kind of a standard text and reference. It's oceanography of the Georgia Strait, and there's a whole chapter on Southeast Georgia Strait, which is Cherry Point.
I need to read that.
And it's very clear that Cherry Point, the primary influence in Cherry Point oceanography is the Fraser River.
Yeah, absolutely. And then the other thing that I've seen is that the temperature threshold between where it's well mixed right north of the San Juans and where it becomes very warm, that seems to move north and south depending on time of year mixing. But it's really interesting.
Thanks. Kim, I think you're introducing the next speaker.
Am I bringing my microphone?
Yes, you are. Thank you. Right. So I just want to remind you all that after this, if you want to stick around after Ian, we'll have all of the speakers from today come up and you can ask any questions you'd like. So thank you.